de estos postdoctorados le contrataron como profesor en la Universidad de Florida, en donde estuvo unos años, del 2015 al 2021, y en el 2021 le contrataron como profesor y jefe de departamento de zoología en la Universidad de Texas A&M. Entonces, Alex ha estudiado cosas súper interesantes de genética de moscas y de peces y nos va a platicar uno de sus proyectos, o nos va a mencionar varios, pero se va a enfocar en uno, que es el que trabaja con Patty en los peces ciegos de aquí de México. Entonces le damos la bienvenida a Alex. Muchas gracias a ustedes por venir y gracias, Alex, por estar aquí con nosotros. Thank you.
All right, I will try not to wander. I have the tendency to wander around, so someone scream at me if I do. But thank you, Gabby and Patty, and all of you for coming. It's an honor to be invited here and to get to talk about my work. My lab's been working for the last almost 15 years now, developing Mexican cavefish as a model to understand how we sleep and sleep differences between individuals. I have another half of the lab that works on sleep and fruit flies, but I thought I'd focus this talk just on cavefish. And what I hope to convince you is that we have a system where we can really take the power of evolution and then use that to understand some really basic things about human health and human disease. And so please stop me at any point if you want me to talk about something in more detail, or if something isn't clear. It's not advancing. Is there? Do it. I can also just say next slide. There you go. Perfect. So I'll show you this picture here. This is the subway station in New York City. It's crowded, probably a lot like Mexico City. And what you might notice is that everyone looks different. But what you can't see is that a third of the people in this picture have some form of sleep disorder, the most common of which is insomnia, trouble falling asleep. And what's really interesting about these sleep disorders that a third of the people have is a lot of them have very different biological causes, even if they manifest in the same way. So two people who have trouble falling asleep could have trouble for very different biological reasons. And so we're really interested in figuring out what the causes are, and that's really the foundation for more personalized treatments in medicine, instead of giving a general treatment that might affect everybody in a non-specific way. There have been lots of human genetic studies that have tried to address this. Making sure it's okay. Okay, but should I just keep going, or should I pause? Okay. Good. Okay. So there have been these very large scale human genomic studies that have tried to understand differences in sleep between people. Some of these have had millions of people, up to 2 million people that they've looked at. And it's been challenging to find genes. And one thing that's common throughout these is there are many different low effect genes that seem to regulate how and when we sleep. And the idea here is there are probably lots and lots of genes that contribute to this process, and they differ between individuals. And so this is really, it's a challenge for sleep medicine. And that's why I've really tried to take evolutionary approaches to address this. One of the nice things about sleep is we can measure it through behavior. And so we might think of measuring sleep by looking at electrical activities in our brains, but there are also very clear behavioral measurements of sleep. And so things like behavioral quiescence, when we're asleep, we're usually not moving. There's an elevated arousal threshold. So a noise that might get your attention when you're awake, you won't hear when you're asleep because your brain is blocking out those noises. And so we can have very good ways to measure sleep through behavior. And these approaches have been used to define sleep in lots of different models across the evolutionary tree. And so it used to be thought that sleep was something relatively specific to mammals. And we now know that insects, and even there's really exciting work now that jellyfish sleep, and jellyfish don't even have a brain. And so that suggests that sleep is maybe a cellular property and not a network property of an entire brain or networks of connections. And so scientists have used this, and including my lab, to find new genes involved in sleep. And this is an example where you can take fruit flies, you can put them in a tube, the fly will walk back and forth, and a little beam there will measure its activity. And then you can quantify when it's inactive and call that sleep. And this approach has been used to find many new genes involved in sleep. But there's a real problem with this approach, even though it's been very effective, and my lab uses this as well. So I'm not saying it's not worthwhile, but all of this work has been done in isogenic animals for the most part. So animals where every genome is the same. In worms and fruit flies, zebrafish, mice, right? We've inbred these animals to take away their naturally occurring genetic variation. But when we talk about issues of human health or genetic variation or the important things that drive evolution, that genetic variation is one of the most important factors. And for that reason, we really started to think we needed evolutionary approaches to get at this question of why we sleep different as individuals and why some individuals have insomnia and others don't. And so we turned to this really powerful model, the Mexican cavefish. And many of you probably don't need an introduction to this, but I'll just give some brief background. These are a really exciting model because these fish are the same species, but they look very different. The cave populations are albino, they've lost their eyes. The surface populations live in rivers and look like normal fish. And the reason for this is they've evolved in very different locations. So this is Rio Choi, where we might have collected, or Patty often collects her surface fish. And this is one of the caves, and you can see how different they are. So in the rivers, there's sunlight, there's photosynthesis, there are predators, and in the caves, there's a lack of these. The cave fish are basically the top of the food chain, and there's very little food. And so we thought these differences might result in dramatic changes in their behavior. These have been studied for a long time, almost 100 years since they were discovered. And in fact, Charles Darwin wrote about cavefish in Origin of Species, not this species, but a different type, an American cavefish, as a really powerful way to explain natural selection. But what's really unique, I think, about Estionyx, these Mexican cavefish, is that there are 30 geographically isolated caves that have converged on very similar phenotypes. And so here you see 4 different cavefish populations that look very similar. They're interfertile, and so we can breed them together, and then we can look to see how traits transmit. And what really excited us at the beginning is that these caves were at least thought at the time to be very nutrient poor. And when I started working on this, I was a postdoc in Justin Blau's lab at NYU, and I was working on how feeding affects sleep in fruit flies. And so I knew at the time that feeding state was a very potent driver of sleep. And so I thought if these cavefish had evolved to be in a nutrient poor environment, maybe their sleep was different. I teamed up then with Eric Dubuis, who's now one of my closest friends, but I didn't know when we started this work. So it's a reason why I think one of the things I think is so special about science is you form these lasting friendships. And he was a grad student in Richard Borowski's lab who recently retired, but had worked in the system for decades. And we wanted to study sleep. And at the time, people weren't really doing this in fish. There was one lab that was studying this at Harvard. It was Alex Shear's lab. And so we got on a bus from New York to Boston. It's 3 hours. And Alex was kind enough to show us how he was measuring sleep in zebrafish. And it was basically to take these young fish, we chose 20-day-old fish, and measure their activity. And so this is an example of one of the first video recordings we made. Also, probably the only time in my career an experiment's worked out the first time I've tried it. And you can see there are cavefish here. This is in the middle of the night, and we're recording through infrared, so the fish can't see it, but we can. And you can see the cavefish are active and the surface fish aren't. And so this is just a really clear phenotype. We were excited. We wanted to quantify this. And so we took 3 different cave populations and surface fish. The surface fish is in black, and it's sleeping over this 2-day period. That's basically identical to zebrafish. I'm not showing it to you here, but if we were to overlay zebrafish, it would be the same. So it's probably normal fish sleep. And if we take these 3 different cavefish populations, you can see they've all lost their sleep. And so we knew we were on to something really exciting. There were very few animal models at the time that had lost sleep and lost most of their sleep. Even the evolution of sleep really wasn't studied very much. There's one person, Jerry Siegel, who's done incredible work in dolphins and whales, and we kind of based this off of his findings. So we've been very interested in understanding why these fish lost their sleep. And that's kind of what my lab's been working on since it started. I'll tell you a few stories about why we think these fish aren't sleeping. And then I'll talk about how I think this cavefish system can be used to study lots of different traits. This is James Jaggard, who's a grad student in the lab. Now he's at Stanford. He's dissecting fish. If you can see this tarp here, this is because he was dissecting in the middle of a major hurricane that hit Florida. This is when our lab was in Florida. And I stayed in the lab to make sure the fish survived. And he stayed because he didn't trust me to take care of the fish. And then he ran experiments throughout the hurricane. But he was really interested in this idea of arousal threshold and whether that is driving the sleep loss. And so you might think there's less sensory input into the cavefish brain because they don't have eyes. But in fact, they've developed all these other senses to compensate for losing their eyes. So they have better sense of smell, better sense of touch. And he thought maybe this was related to the sleep loss. And we know this is an important driver of sleep. This is an old paper from Paul Sternberg's lab in worms. And he shows there are actually neurons in the brain, 2 neurons in this case, that block out incoming sensory stimulus during sleep. And we know this happens in humans as well. Okay. So we asked what happens if we disrupt sensory input to cavefish. Just to show you kind of the sensory changes, this is different work from our lab, but you have a surface fish on the left and it's attacking its prey, Artemia. And you can see it does so straight on because it's using its eyes. It's a visual hunter. And this cavefish does something very different. It's using its lateral line. It's bending. And in fact, if you ablate the lateral line, the surface fish is fine and the cavefish can't hunt. And so it's developed this expansion of the lateral line. You can see that, I don't know how well you can see this here, but we're staining for these lateral line neuromass, and there are more of them in cavefish than surface fish. And so we asked whether this could be contributing to sleep loss. Can use very simple approaches to just ablate the lateral line. So we bathe the fish in an autotoxic antibiotic, gentamicin. It kills off the hair cells. They grow back later. But that ablates the lateral line, as you can see here in surface fish and cavefish. And we can say, what does that do to sleep? And what we saw was it actually really surprised us. So there's very little effect on sleep in surface fish. These are the ones with the ablated lateral lines. But if we ablate the lateral line in cavefish, sleep is almost completely restored. So this lateral line, the sensory input from the lateral line of fish is driving their sleep loss. You might say, oh, these fish, they can't sense anything, so they're not moving at all. But this graph is how active they are when they're awake. And there's no difference. There's no effect of the ablation. So it's actually the lateral line input is preventing cavefish from going into a sleep-like state, and we restore sleep when we give it back. I'll skip this. So we had this idea then that basically we have this lateral line that's wake promoting, right? It's driving wakefulness in these cavefish. It must be doing something in the brain in order to keep them from entering a sleep-like state. And we wanted to know what that is. The first place we looked is the main wake-promoting neuropeptide in mammals, hypocretin or orexin. And so this is very famously found by Emmanuel Mignot, who had these narcoleptic dogs. And so the dogs, every time they got excited, they would pass out and go to sleep. And he mapped this about 25 years ago to a mutation in this hypocretin receptor. Shortly after that, Jerry Siegel's group published that humans with narcolepsy have a loss of their hypocretin neurons in their hypothalamus, which he attributed to some autoimmune function. And then work from a number of labs, Alex Shear's lab and Lior Appelbaum's lab has shown that if you ablate the hypocretin receptor neurons, you increase sleep in zebrafish. So it's a highly conserved wake-promoting neuropeptide. Our thought was that perhaps there is increased hypocretin neuropeptide levels, and then that's going to drive wakefulness in cavefish. If we look at the levels of hypocretin in cavefish compared to surface fish, they're higher, and they have a higher number of cells. Am I good, Patty? All good? Okay, awesome. And you can actually see that here. So here we're taking a section of the brain, and we're staining it for this neuropeptide hypocretin. And you can see there are more cells in cavefish, more of these wake-promoting cells than surface fish. But this doesn't, just because they have more of them, doesn't mean it's actually causing them to sleep less. And so we wanted to really show we could functionally manipulate this system and that it's causing the sleep loss. And one of the main drugs that's on the market now, it's very controversial for people with insomnia, is a hypocretin receptor inhibitor as of about 5 years ago. It's controversial because it's unclear how effective it is, but they designed this drug in a really interesting way. And they basically took the crystal structure of the receptor and then they designed an antagonist for it. And so we had good ways to pharmacologically manipulate this receptor. And so if we bathe these fish in this inhibitor of hypocretin, there's little effect on surface fish, but we restore sleep in cavefish. We then use this approach, morpholinos, to block the translation of hypocretin to lower levels in cavefish, and it increased hypocretin levels. And then James Jaggard, who is the grad student, performed what I think was just a really gutsy experiment. And I tried to talk him out of this, where I said, we have 2 ways of confirming this. We don't need a third. And he insisted on doing this, which was he ablated the neurons genetically. So he genetically expressed this toxin, botulinum toxin, in the hypocretin neurons and restored sleep. And this was just a really important experiment for my lab and maybe the field, not because it told me anything specific about sleep, but because I think it convinced us that we could use these genetic approaches that had previously largely been limited to fruit flies or mice or zebrafish in this model of evolution. And then we would have the strength of a comparative model of evolution and a genetic model. I should say these changes that we see aren't strictly at the level of transcription. And so if we look at variation in the hypocretin receptor, we see polymorphisms in the hypocretin receptor. And so we think in the wild, evolution has done a trick for us where they've produced more hypocretin and they've changed the receptor to be more sensitive. And we're currently making the gene swaps to see what these receptors do. We can model these onto the structure of the human hypocretin receptor. And at least 2 of them down here, we're very confident affect the signaling, the activity of the receptor. So we think there are multiple ways in which this pathway has been enhanced evolutionarily to drive sleep loss. And I think this shows the power of evolution. So if we were a drug company, we might spend a long time taking this receptor that we know is involved in sleep and randomly mutating it and looking to see which mutations affect its signaling properties. But in this case, we didn't need to do that because evolution had done the trick for us. And so we just needed to see where the changes occurred within these naturally occurring fish that existed in the wild. And so we think that this wake promoting input from the lateral line is driving elevated hypocretin levels. I didn't show you, but if we ablate the lateral line, hypocretin levels drop. And then this outputs to dopamine and norepinephrine signaling and other mechanisms that regulate sleep. And so the long and short of it is we've gotten at some of the neural and genetic mechanisms that are driving sleep loss in cavefish. But there are lots of questions that are still unknown. And so the lateral line conveys this information into the brain. It's weird looking at myself up there. The lateral line better conveys information. It's like in the old days when listening to your recorded voice is also unpleasant. Okay, lateral line conveys information to the brain, right? How does it convey that information to the brain, to the sleep centers, and how is that processed? That's a big question, and we don't know it yet. I told you that there are these major changes in the hypocretin receptor, but we know that with this incredible level of detail in zebrafish, the neuroanatomy of hypocretin neurons, have those changed in cavefish? Do they project to different brain regions? Open question. I think one of the main strengths in zebrafish neuroscience, I would argue what makes it one of the best models for neuroscience out there, is this ability to do whole brain imaging. And so in the zebrafish larva, there are about 100,000 neurons. They can image from every single one of those neurons at the same time and then figure out how neurons are connected. And if we could do that in a model of evolution, it would just be incredibly powerful, not only to understand how brains evolve, but maybe understand how our brains might all wire differently. And then everything I talked to you about is in this one population, the Pechone population, but there are other populations of cavefish that lost sleep. And is it through the same or different mechanism? And just to give you a hint, when we ablate the lateral line of other populations that we've tested, there's no effect on sleep. And so that lateral line phenomena is specific to Pechone. There are other mechanisms of sleep loss in these other species. They all have elevated hypocretin levels. And I think that highlights the strength of this system. You can have very different ways at getting to the same result. So I just want to tell you a little bit about how we're trying to develop these questions. And a lot of this is collaborative with the community. It's not just my lab, but I really think this as an evolutionary model has potential to be as powerful as any of the commonly used genetic models like fruit flies. And so hopefully I can convince you of that. We're trying to map traits, basically crossing surface fish and cavefish and creating F2s. And then we can look for how much they sleep. And then we can try and find the locations in the genome that associate with that variation in coding sleep. We did this in a very crude way in adult fish. This was an underpowered study of a couple hundred fish. And we found basically one region in the genome that associated with elevated locomotor activity. But we knew at the time this was really insufficient. This was when Masato Yoshizawa, who now has a lab at University of Hawaii, was a postdoc with me. So we're doing this again, and we're doing this with thousands of fish this time. It's a large collaborative effort with a number of labs, Nick Roner, Joanna Kowalko, Suzanne Magaw. And the idea is just to take hybrids. And so we take these F2s and we breed them together, and you get fish that look very different. So these three are albinos, these are pigmented, and we say which ones sleep, which ones don't, what genetic variation is associated with that. And so one of the things we found very early on, before we even did our genomic sequencing, is that the albino ones slept less. And so here's the average sleep for the quarter of the fish that are albino. Here's the pigmented ones. This is a very bizarre phenomena because it's always thought that the albinism was just completely different from these behavioral traits. There's work suggesting, so we know the gene that causes albinism, at least in Pechone. It's this OCA2 gene. It was cloned by Cliff Tabin's lab, by Meredith Protas and Cliff Tabin's lab. And then there's really interesting work from Helena Bailanzija, where she shows that when you mutate this OCA2 transporter, that's essential for transporting tyrosine into these melanosomes, you get elevated levels of tyrosine. And that's important because tyrosine is a precursor for dopamine, which is wake promoting. And so we thought maybe this was a mechanism, maybe the albinism all along was to drive a behavioral phenotype. We never would have known that. So Joanna, who was my colleague, and sadly isn't anymore, we had labs next to each other at FAU, used CRISPR, and she created surface fish that just had a deletion in this OCA2 gene. So these are fully surface fish, only they're albino, and they slept less. And so we could validate a role for this gene in sleep. And now we're trying to take this approach, and we're going to look for genes throughout the genome. We're doing this with Suzanne McGaugh, and then we're going to try and validate them. We also measured feeding, we measured body fat levels, we measured all sorts of other traits. And so hopefully we can find genes associated with all these different processes. Just to give you an example of how we might do that, here we looked at gene expression over the circadian cycle. And you can see here, we're taking multiple measurements, and there are lots of genes that cycle in a circadian fashion in surface fish. You can see that pattern there, and they're absent in cavefish. And one of the genes we found that had lost its rhythmicity was this transcription factor, RORCA. And when we use CRISPR to knock that out, daytime sleep is normal and nighttime sleep is lost in surface fish. And so this is maybe not that exciting in terms of what it tells us about the biology. We already knew this was involved in circadian rhythms, but it just shows we can find genes through mapping studies, and then we can functionally validate them through gene editing. And I think that's a really powerful approach, whereas like in the fruit fly part of my lab, we just take a list of 1,000 genes and try and screen them over the course of a year. Here again, we can use what evolution has given us and then target those genes. Here's an example of how we're doing that. And so we're injecting CRISPR into the eggs, CRISPR guides, and we're knocking down genes in surface fish and cavefish. And these are genes where the genetic analysis suggests they have markers of selection or they're transcriptionally dysregulated. And then we can look for ones that sleep or don't sleep. And we see some pretty striking phenotypes. Like this is a protein tyrosine phosphatase. And when we knock it down in cavefish, there's no effect. But when we knock it down in surface fish, there's a reduction of sleep. And here's another one. This is a G protein subunit. This is very preliminary, so don't hold me accountable for this data. But when we knock this down in surface fish, there's very little effect, but there's this giant increase in sleep in cavefish. And it just shows the power of basically screening in both cavefish and surface fish, because you get different sets of genes depending on the model you look at. So I think a main thing that confronts this field, and if we really want these fish to be kind of the zebrafish of evolution, we need to develop the genetic tools. And I mean, there are just incredible things you can do in mice. We can look at specific neurons and how active they are. We have this really detailed understanding of their brain structure in their development, and we can basically knock out genes at will. Same for zebrafish and flies. And this isn't because, evolutionary biologists don't work as hard. It's because there's been, 60, 100 years of concerted effort developing this single mouse species as a model organism, right? And you have thousands of labs working on this and billions of dollars in funding every year. Whereas most evolutionary models, it's only a handful of labs. And so we're hoping as a community, we can come together and develop some of this in cavefish. And then we'll have two things. We'll have this level of resolution and the power of comparison. And so the first step in this, and I'm biased because my background's in neuroscience, so a lot of what I developed were the neuroscience tools at first, but I really wanted to be able to label different neurons within the brain. And so here, for example, we took the promoter of hypocretin and we stuck GFP in front of it, green fluorescent protein, and you can see the level of resolution. We can see individual axons and dendrites coming off the neuron. Whereas when I showed you the antibody stain for hypocretin before, it was like a green dot that was the cell body. And so now we can really look at how these neurons have changed between species. We can label whole brain regions. And one which I want to talk a little bit about is this GCAMP line, because I think it's particularly powerful. And so this is a calcium sensor. And so when neurons are active, calcium rushes in. And so this is a protein that glows brighter when calcium's in the cell. And so you can use it to know when neurons are active. And I'll just show you this. This is the retina. And so we first characterized the visual system of cavefish because we thought that's where the most obvious differences would be. And so this isn't the retina, this is the optic tectum where the processing occurs. And if you look, you can see these cells flashing on and off. Those are the neurons being active. And we'll flash a light. And sometime there should be a light that comes on. And then we can look at the response to the light. We can look at the response in the darkness, and we can see how visual processing has changed within surface fish and cavefish. To give you an idea of the analysis, what we're doing is we're breaking down each one of these cells in the tectum, and we're looking for these blips of calcium, and we call that a neuron firing. And we can do that across every single neuron here. We can then look at the probability that if one neuron fires, another neuron fires. There's an enormous amount of data that's wrapped into this video that we actually don't understand. And that's why we were collaborating with Herman Sombre. And so what did we find? We found the number of neurons was lower, just the number of cells in the optic tectum in both cavefish populations that we looked at. This has been basically known for 40 years. So this was not surprising. It was just confirming our analysis worked. But what was surprising is that in darkness, so with no light, there was just as much activity in the optic tectum. So cavefish had input from their eyes. At one point, that input was lost, right? Because they don't have eyes. And so you'd think the area of the brain that received that input might stop working. But it's not, it's just as active. And if we look at the correlation, if one neuron fires, is another neuron likely to fire? We find that there's just as much correlated activity. And so what this really suggests to us is that this area of the brain that should have undergone disuse because they lost their eyes has been repurposed for some other function. And so we're interested in figuring out what that is. Our preliminary data suggests that if you touch the fish, this area lights up. And so we think this area of the brain that's visual processing now in cavefish is for touch processing. And here's just an example. We can classify these neurons in different ways. And so within the tectum, there are neurons that turn on when the light comes on, or turn off, or they only come on when the light goes off. And basically there's a loss of most of these in cavefish. There are many fewer cells than in surface fish. We just, because we study sleep, we wanted to apply this to sleep. About 2 years ago, Philippe Morin at Stanford, his lab published what I think is a really important Nature paper in zebrafish, showing that they had sleep stages just like mammals. So they had slow wave sleep, they had REM-like sleep, and we wanted to know whether these were present in cavefish and surface fish. And so if you look at this video here of a surface fish, the activity is asynchronous. They're coming on and off while they're awake, but it's synchronous activity in the sleeping fish. They all come on at the same time. This is similar to our brains when we're asleep. We get synchronized activity. And so we can plot this. You can see the lines are all squiggly. So each one of these is a single fish, and each one of these lines is a neuron. And you can see there's no synchrony in surface fish or cavefish when they're awake. They become synchronous when they're asleep. And if you look at this carefully, you can see they're more synchronous in cavefish than in surface fish. And so James Jaggard, this is my old grad student who's now a postdoc with Philippe and is heading up this work, has this hypothesis that this increased synchronicity is actually what allows the cavefish to sleep more efficiently. And that's why they're sleeping less. So it's not that they're actually insomniacs, it's that they're more efficient sleepers. we want to get at what the anatomical differences are between these fish. So the cavefish brain just looks different than a surface fish brain. These were the immunostains I showed you. Again, they don't look great, but zebrafish have this incredible resource where they basically develop computational brain atlases. And so they took the images of hundreds of brains, then averaged them together to call it a virtual brain. And they get this incredible resolution. And so this is a paper from Alex Shear's lab. We thought if we could do this in surface fish and cavefish, we could know how every brain region changed between the populations. And so the way we do this is we take confocal stacks of the brain. This is a 5-day-old fish. And so here are two different brains. One is red and one is blue. And we basically average them together, right? And then imagine doing that hundreds of times over. And then we add immunolabels to define specific neural circuits, and you build the brain atlas that way. And so here's an example of what you might end up with major brain regions labeled. And so if you do that, you can see, just looking at it, the brains look different. So there's a smaller telencephalon in this region here and the cave populations than surface fish. But we were really interested in understanding at a very fine scale how the neuroanatomy changed. The idea, we'll build these videos, and then we'll kind of use that to quantify differences. And we can do this looking at neural activity as well. So let me skip over this. And I'll just say that that was the first version of the Brain Atlas, and we now have even better versions out. And so what we did is, this paper is under review at eLife, and Eric Dubuis is the lead author, but it's basically the second version of our atlas. And we took this program called Cobra Z. And so what it's doing is we're using the zebrafish brain as the atlas. We're quantifying all the brain regions in osteonics to that atlas, and then we're extracting them individually. And so before, we kind of manually annotated the regions, and now if we put a fish under, the computer can tell us the size of every single brain region. So here, each brain region is a different color, and we can know how the brain changed in size. And what's especially exciting about this is it works for surface fish and cavefish. And so now we can generate F2 fish, these hybrids, and we can look to see in every individual fish how their brain differs. And the Allen Institute is trying to do this in humans, but obviously human experiments are much less controlled, and you can't go through, well, you can go through thousands of humans at an incredible expense in MRI, but we can really ratchet this up now. And to give you an example, here we have the surface fish. So this is the pallium, which is an area that we think is involved in sleep. And surface fish have a smaller pallium, so it's less than 5% of their brain volume. It's larger in cavefish, and the F2s have an intermediate phenotype. And so we can ask a question like, are these cavefish with the larger pallium shorter sleepers, right? And that we would have been largely inaccessible. And so the real long-term goal is to go from behavioral variation. So we'll take these fish, we'll measure behavior in all of them. We'll start with sleep because that's what I like, but we can measure any behavior. We can measure feeding, and then we'll look at their neuroanatomical variation by putting them through this brain neuroanatomical analysis, and then we'll sequence. And if we do this, an individual fish will be able to understand the link of genetic variation, neuroanatomical variation, and behavior. And that's just, it's not even a tractable question in mice or zebrafish or flies, but it's something you can only get at if you're working with existing genetic variation. And just to give you an example of this atlas, because I think it's especially exciting, we can basically take individual brain regions like the diencephalon, and we can drill down on them. And so here's the difference. The diencephalon's larger in cavefish. But if we look at the dorsal diencephalon compared to the hypothalamus is larger in cavefish. The dorsal diencephalon's larger in and surface fish. And so we can really get a high level of resolution. We can also look at in F2s the relationship of every brain region to one another. So these brain regions are developmentally specified, right, by transcription factors or axon guidance cues. And there's a lot of interest, across biology in how the brain develops. And so we can say if one brain region is larger, is another smaller. And if we cluster them, so here we're just clustering the relationship between every brain region with itself, with other brain regions. And we start to see very interesting patterns emerge. So sometimes it's brain regions that are right next to each other that are correlated. That makes sense. Sometimes a brain region very far away is related either positively or inversely to the size of a brain region on the other side of the brain. And that's probably because there are shared genetic neurodevelopmental pathways that specify the development of those brain regions. And so we also think this resource could be really effective for understanding basic principles of brain evolution. And our goal is to put this out there. So we're just going to make these atlases, we're going to collect brains, and then we're going to put it up as a resource for different research groups to use. Just want to take a step back and give you one last justification for using this model. These are really weird fish. And I've talked mostly about sleep, but they have all sorts of other fascinating traits. And so Nick Roner's lab at Stowers has shown that these cavefish are diabetic. They have a mutation in their insulin receptor. They're hyperphagic, so they overeat, and they have deficits in heart regeneration. So zebrafish and surface fish are very effective at regenerating their heart if you damage it, but cavefish can't do it. And these are all traits that you'd expect would be just terrible for the fish. These, diabetes, sleep loss, hyperphagia are all associated with neurodegenerative disease in humans, as an example. But these fish seem healthy. And I haven't had a lab long enough to know, but if you ask the old timers like Cliff Tabin or Bill Jeffrey, they'll tell you these cavefish can live 30 years, just like surface fish. And so we talk a lot about these cavefish as a model of disease, like insomnia or diabetes. But another way of looking at it might be, are they a model for resilience, right? Why are they healthy despite having all these traits that would make just about any other animal sick? And I think that's especially exciting because lots of people study disease, many fewer people study resilience. And in some respects, all of evolution is resilience, right? We have 4 distinct parts of our department at Texas A&M, and one of them is biological resilience and another one's evolution. And someone said in a meeting the other day, hey, isn't all evolution just resilience? And I think there's good argument for that being the case. So I want to start asking how they're healthy. And looking at longevity isn't probably a great way to do this because they live for 30 years. So we need other markers of them being healthy. And then I just wanted to end by saying some work we want to do bridging their natural habitat, how they act in the wild with what we're doing in the lab. And so this is an example of a paper that I was really excited about. Patty collected these fish from Chica, which is a special cave because it's a hybrid cave. And so they're a mixture of surface fish and cavefish. She was kind enough to host James, a graduate student, in her lab. And they measured sleep in these wild caught fish. And then Suzanne looked at their genomes, Suzanne McGaw at Minnesota. And they found basically that in 2 different pools of Chica, the same cave, there were sleep differences, with some in this first cave being more like surface fish, and in the second cave being more like cavefish. And what was really interesting is they found an association between variation in this cryptochrome 1, this thought to be magnetosensing circadian transcription factor that has this single amino acid substitution here, and how much they slept. And so there's this association, but then when they looked for this association, they found that it was present in other cavefish from all over the world, like Somalian cavefish from Africa, Chinese cavefish, even some burrowing mammals, like naked mole rats. And so to me, this is still an insane result, right? How could this one genetic change be associated with animals that aren't even remotely related, but it's there in the genomic data. And I think we never would have known that unless we'd actually pulled fish out of the caves. By we, I mean Patty, and then phenotyped them. We've been setting up slowly to look at what the cave environment is. And I should say I have two collaborators on here, but I think this is a real community effort. And the question is, what is it? What's different about the caves across seasons, across individual caves that's driving these evolutionary phenotypes? So Jessica Hua is a community biologist, and Maggie Osborne's a biogeochemist that's interested in microbial communities. And we've been taking samples of water out of the caves and looking at the microbial life. What Jess's goal is to set up these mesocosms. So imagine these small containers that recapitulate the cave environment, and then to look at behavior within them and manipulate the behavior, manipulate the environment and see what's driving these behavioral changes. This is an example. You can see, so this is the microbial communities, and you can just see they're very different within the 3 cave populations, the 3 cave readings than within the river. And then there's a large community effort now to implant these probes. And here you can see surface fish right next to the probe watching over it. And I think these will tell us, how the water chemistry changes over the course of the year, and allow us to better perform experiments in the lab. And I just wanted to end highlighting this really large effort for the community that Patty really played an instrumental role in. This is Suzanne McGaw, Minnesota, and Andy Glusenkamp, who's director of conservation at the San Antonio Zoo. And they're actually in the cave catching fish, recording them to measure sleep in the wild. And they can get these nice videos. This is a surface fish. And now we're actually going in and we're analyzing the data, which has been a real challenge, but we can do it. And so we're going to see whether these sleep phenotypes we see in this lab setting are present in the wild, and if they're there throughout the year. I just want to give credit to the lab. And so we had this really interesting situation that I think in Florida, where there were 3 of us, 3 cavefish labs right next to each other. So Eric DeBui's lab and Joanna Coaco's lab. Eric's still there. Joanna left to go to Lehigh. I left to go to Texas A&M a few years ago. It's very sad, but we still collaborate incredibly closely. And it was basically the chance for the 3 of us with very different expertise to work together. We mixed up all the lab space. Almost all the grad students were co-mentored. And I think a lot of this work wouldn't have happened without that environment. We're a smaller group now in Texas, but we're excited to keep working on this stuff and collaborating with the community. So thanks so much.
Muchísimas gracias. Una disculpa a la gente del Facebook porque hubo ahí una confusión con la liga, pero estamos listos para tomar preguntas. Muchísimas gracias. Thank you so much, Alex. It's fascinating. Entonces, estamos listísimas con el micrófono para recibir sus preguntas. No.
Hola. Hola. Aquí está. Hi. So many questions. Okay, so I have 5 questions. So the first one, there is a guy at Stanford, I think his name is Andrew Haberman, and he has a very famous YouTube channel. That's how I know about him. He's a neurobiologist, but he specializes in ophthalmology, I believe. So I was thinking about how he's very intense about the effect that light has on our circadian rhythm in humans, right? And he's always promoting that sort of to manage depression and not feel motivated and all of this, you should get like 20 minutes, face the sun every day, right? Around like 8 a.m. So I don't know much about the fish, but and you mentioned a couple of times that they don't have eyes, right? But so that's one of my questions, like they don't have anything that resemble an eye. And also it's very interesting how you landed at the connection of how they are albine, but also how the correlation between albinism and sleep loss, because that could be also connected to this, well, sensory to light, right? Again. So that was one question, because I was really curious about your interest in the lateral line. Like, is there any evidence that lateral line has any effect on light, like capture? The second question
Wait,
is—
can I answer the first?
Sure,
And then, because
sure,
otherwise
sure.
I'll forget them. And then no, so I think no, I think it's a really good point. We don't know. So there's a great paper from Masato Yoshizawa where he shows the pineal gland is expresses rhodopsins in cavefish. And so I think there might be the possibility that they're still, well, we know they're still sensitive to light, because if you look closely, when we look at their sleep, we see some diurnal activity. And so they know the light's there. And so it might be through these deep brain photosensors, opsins that are still there, but not the eye. And so it's a great question.
I also wonder if you force them to receive more light, not to be able to swim away from the light to darkness, but in an experimental setting. And then you measure sort of their activity, if you can get a glimpse of a circadian rhythm or something like that. I don't know.
Yeah, I mean, we really, circadian rhythms for just technical reasons are harder to study in fish than they are in, say, fruit flies or mice. But this is something we really want to look at. There's some cavefish that don't have mutations in OCA2, but have reduced pigmentation. And one of the interesting things that happens is if you put them in bright light, some of their pigmentation returns. And so yeah, all of these are great ideas. We just haven't done it yet.
Another one was about, how do you really know the behavior of the cavefish? Because you said you kind of take cues from their behavior to assess whether they are sleeping or not, right? But they might, just because they are cavefish and they are weird, have different behaviors than a surface fish. So whatever you are interpreting as a sleep in a surface fish might not be sleeping in a cavefish.
Yeah, or they might be having these micro sleeps, right? And so we did more work than I showed to kind of really try and look at the details of sleep. But I think the real next step is to do these brain recordings, and hopefully that will show us better resolution.
Okay, I think I can live without the other two, but I have a third one, and it's about, so when you show the crosses, you got 3 out of 9 albine cavefish, right? So it looks like a recessive sort of Mendelian way of inheritance. And they live in a cave. I wonder, like, how much predation do they suffer? Because, and then later you show a lot of variability in the responses, in the cavefish responses. And to me, that makes a lot of sense if they don't have a lot of, natural selection pressure, and they are like 1/3 of, like a recessive thing, and they are so ill. I mean, so they have so many issues, right, with their health. So they seem like a suboptimal phenotype. So I would expect a lot of variation, a lot of intraspecific variation might be allowed in those populations, right? Because, so I wonder about their natural, is there any natural selection on the cavefish? That's the question.
So Patty is probably better positioned to answer this than me, but I'll take a stab and then you can correct me. So I think that this question about the mechanisms of evolution in them, like whether it's neutral evolution or whether it's being driven by selection, is something that people have had huge arguments about in our cavefish meetings. I tend to think that the evolution happens so quickly that there are probably strong selective pressures. And there's actually, there's much more homozygosity in the cavefish because there are these bottlenecks, right? And then so it looks like they're under very strong selection, but the predation, I'm not sure about. There are constantly surface fish that are being swept into the caves. And then I don't know how many of them breed or.
Otra pregunta.
Thank you for the presentation. One question that I had was, do you observe changes, well, differences in drought in the hybrids? Like, is there something that is unexpected in the hybrids on how they grow or how much they resist diseases or pathogens? Perhaps will be associated with the sleep loss or.
Yeah, so we've only looked at sleep and feeding, I think, in them. Well, and fat stores, which are conveyed by a single locus. So I'm not sure, but I think it gets to the broader question of doing some sort of competition assays, right? Or looking at them, doing some experimental evolution in hybrids, which I would love to do, but we haven't done.
Thank you.
Yeah.
Hi. Well, my question is, what are your thoughts on an experimental design that can distinguish between process S, the homeostatically regulated sleep, and process C, which would be circadian regulated sleep, either assessed behaviorally or with the new approach, with the neural mapping approach?
Yeah, that's a great question. So there's a theory, kind of, it's probably the most long-standing theory in the sleep field, that there are two components to the brain. There's the circadian process that tells you when to sleep, and there's a homeostat that tells you how much to sleep. I think it's going to be more complicated. I didn't show it, but we had experiments where we sleep-deprived cavefish, and they make up their sleep. And so the homeostat is intact, but their basal sleep requirements are lower. To some degree, their process C is intact because they sleep more at night during the day, but not by a lot.
Like cavefish weren't able to be entrained with light.
Oh yeah, I should say that. So they can't, yeah, there's no free running circadian clock. So if you put them in the dark, they don't maintain their rhythms, but they sense light. So maybe its process C is gone, is the way you would interpret it in that context. There's a wonderful paper in Somalian cavefish where they show that you can't entrain them with light. It's a totally different species, but you can entrain them with food. And they have some ecological reasons for why that is. And so if you give them food the same time every day, and then put them in an environment without food, they'll still maintain that activity pattern, but you can't do it with light. Whereas almost all animals, light is the strongest driver of our circadian clock.
Thank you very much.
I will take one of the questions from YouTube, and I think somehow you have already answered part of it. But Victor Sanchez Cordero says, excellent seminar. Have you thought to put surface and cavefish under stress experiments and see if they change sleeping patterns? And by stress, he means food deprivation, predation pressures, etc.
Yeah, so I think I think a lot of these questions are getting at something we know we need to do more of, which is to mimic the natural environment. And so the one way we've done this, which actually is going to complicate everything I've told you, is we starved these fish and we looked at the responses of surface fish and cavefish, and we find the opposite response. So the surface fish are like the fruit flies we study in my lab, and if you starve them, they sleep less. And that's probably because when they're hungry, they're going to go out and forage for food. The cavefish actually increase their sleep when you starve them. And my sense is it's because that's telling them it's the dry season, there's no food around, it's time to conserve energy. And in fact, Nick Rohner's group had a paper recently in PNAS where they showed the muscle of the cavefish when you starve them actually looks like it's hibernating. And so it might be that a lot of this sleep loss phenotype we're looking at is because our animal protocol says we have to feed them every day, so we feed them every day. And that's telling these fish that this is the wet season and food is plentiful and it's time for them to stock up. And so they shouldn't sleep a lot because they should forage. But but they might have a dry season where they're actually very inactive. And the only way we're going to know that is by going down into the caves, looking at their.
Yeah, I have a couple of questions. Okay. So my first question is from an evolutionary perspective, having tested before, which is the first response from cavefish related to sleep lost when they enter to the caves?
Yeah, that, I mean, I think that's getting well beyond what we know, but in the area of what we would love to do. And we're not going to be able to do that without Patty or the help of some of you here who, are interested in getting into the caves. I'll just say it was really hard doing these. I mean, we're still working on the analysis. So the video was hard because they're fish and they move around, right? And so a lot of it is going to be figuring out the best way to do these experiments. Do we pull them out of the caves and at least we have wild caught fish and measure them in a controlled setting here at the university? Or do we actually try and do these, probably really informative experiments that are really hard in the caves?
The second question is, since they are wild caught fishes, which are the husbandry conditions used to get the F1 offspring? They are special conditions for in-lab reproduction of the fish?
Oh, yeah. So we've been really working on this. This is one of the reasons. So zebrafish have like a 3-month generation time, and it's because it's just been really optimized by so many labs. And so we're working on that. And we have now special diets we can feed them to accelerate the growth of the fish. And then we have a diet, basically a high-fat diet that we give them that promotes breeding. We were worried that if we did that we were going to change all their other phenotypes that we've been studying for so long. It doesn't look like it does. So now we think we have generation time down to 4 or 5 months. It's still long, right? I mean, the fruit fly is 10 days.
No, acá había atrás está Carlos desde hace como una hora.
Great. Thank you so much for your talk. It was incredible to see neurobiology and evolution combined. We usually get talks on systematics and taxonomy, so that's fantastic. And I was wondering, you mentioned that the lifespan of these fish is 30 years about, but you also mentioned that the changes in phenotype can be very, very quick. So I was wondering, and especially looking at the phylogeny where you have this independent events of converging phenotypes for caves, how fast does it really get in evolutionary time for a population of surface fish to evolve into cavefish?
Yeah, this is one of the more controversial areas of the field now. I think I would say a few 100,000 years is probably what the molecular genomic data suggests. There's one paper that uses modeling and suggests 10,000 years for Petchone Cave. I'm a little skeptical of that because the models they use are guessing at some of the variables, right? Like the starting population size. But still 200,000 years is pretty quick, I think, for those massive changes.
¿Listo? Bueno, chicos, primero le vamos a dar un aplauso a Alex y después no se vayan porque tenemos anuncios. Alex, thank you. Va a haber un par de eventos con Alex, pero Patty se lo sabe de memoria.
Bueno, ahora ya, creo que ya. Hoy tenemos el primer evento es pizzas con nuestro ponente y entonces habíamos sugerido que para los estudiantes que quieran tener un momento de plática con Alex, a la 1:30 en la sala de usos múltiples podríamos estar todos juntos ahí para comer pizzas con él. Y el día de mañana a las 10 de la mañana él va a presentar en la Facultad de Ciencias en el Auditorio Moscali
una serie de programas en donde se puede aplicar para tener intercambios con la Universidad de Texas A&M. Entonces, para todos los estudiantes que quieran tener una interacción hoy con Alex un poquito más relajada y poder continuar hablando de su seminario, hoy a la 1:30 ahí en el auditorio de la sala de usos múltiples. Está aquí en el Instituto de Biología en la sede central, ¿sí? Mañana para los investigadores del instituto estamos convocándolos para que puedan estar con nosotros y poder establecer algún tipo de colaboración, algún tipo de intercambio. También a la 1:00 PM en la sala de audiovisuales, en el pabellón, se les convoca para que puedan estar ahí a la 1:00 PM. Entonces sería muy importante, si tienen tiempo, que estén ahí también. Y creo que esos serían todos, ¿verdad? Muchas gracias y gracias, Alex, por. Thank you, Alex.